Okay, now once you've gotten to the point where you understand that there are multiple carcinogens that can damage DNA, then the next point that you have to understand is that there are particular regulatory systems that when damaged will certainly increase the risk for tumor growth and progression. And so the next discussion that we want to have concerns these key regulatory systems. So the three key regulatory systems that we're going to discuss are number one, the proto-oncogenes, number two, tumor suppressor genes, and number three, regulators of apoptosis. These are three key systems that can be disrupted by carcinogenic agents. And these are very high yield, very important. There's a significant amount of biochemistry that goes into them. So what we're going to do is we're actually going to discuss each of these in the subsequent slides. All right, what are proto-oncogenes? Proto-oncogenes are genes that are essential for cell growth and differentiation. All right, and when mutated, so all our cells normally have proto-oncogenes, and those proto-oncogenes are necessary for cell growth and differentiation. However, when a proto-oncogene gets mutated, then it becomes something called an oncogene. And oncogenes, what they do is they lead to unregulated cell growth. So oncogenes lead to unregulated cell growth. Here are the key categories of oncogenes. The categories of oncogenes include growth factor receptors, signal transducers, and cell cycle regulators. I think the best way to understand this notion of oncogenes is to go back and just remind ourselves of normal cellular growth. And so I'm going to go ahead and generate a whiteboard here and just draw you a simple picture to remind you of something. Let's pretend that this is a cell, okay, and this cell has a growth factor receptor on its surface. Now, when this cell needs to divide, and there will be times when a cell needs to divide, either because it's constantly turning over or because there is some sort of repair or regeneration, etc. When this cell needs to divide, what happens is that growth factor comes in, and growth factor binds the growth factor receptor. And growth factor binds the binding of growth factor to growth factor receptor sends a signal, and this signal is carried by something called a signal transducing molecule. And so a signal transducer will carry the signal to the nucleus, and within the nucleus, cell cycle regulators and nuclear regulators will get activated, which will then cause the cell to cycle through G1S, G2, M. And that's the normal cell cycle. All right. So this is sort of the normal growth sequence, and each of these steps involves oncogenes. So oncogenes include, or why don't we say, because we're discussing the normal, each of these steps involves proto-oncogenes. So one type of proto-oncogenes would be the growth factor, another type of proto-oncogene would be the growth factor receptor, another type of proto-oncogene would be the signal transducer, and then another type of proto-oncogene would be the cell cycle regulators. So this is sort of the normal. Now, in each of these cases, mutations in any one of these steps or any one of these subtypes or categories of proto-oncogenes can result in the formation of an oncogene and can then be associated with formation of a tumor. And that takes us then to this table, which is present within your text. And again, it's a fairly dense table, and so it might be easiest for you to follow along in your text here and then to write in the margin some of the notes or some of the points that I may make. So let's take this step by step. All right, the first category of oncogenes are growth factors. So that's way up here at the top of this table. And one of the classic examples of this is astrocytoma, which is a tumor of the astrocytes of the brain, which are a type of glial cell. All right. Now, I don't want to get into the details of what an astrocytoma is here, but I want to highlight to you that in this particular case, what happens is that there is overexpression due to mutation of this growth factor, platelet-derived growth factor B, and platelet-derived growth factor B gets overexpressed, which then drives the formation of an astrocytoma. So let me do this via illustration. So let's pretend that this is a cell, this is an astrocyte, and let's pretend that this astrocyte simply has a platelet-derived growth factor receptor. Okay? And normally, when platelet-derived growth factor binds the receptor, the astrocyte then divides. Now, what happens in this particular case is that you get a mutation so that the astrocyte begins to overproduce platelet-derived growth factor. And if the astrocyte begins to overproduce platelet-derived growth factor, it can actually then sort of swing around and hit the same cell through this signaling mechanism, which then results in overgrowth of the cell, i.e., the extra platelet-derived growth factor keeps hitting the receptor. The receptor then sends signal to the nucleus. The nucleus then recognizes that it needs to divide, and so this cell grows. And this becomes sort of a vicious cycle in which the cell keeps growing in an unregulated manner and eventually produces an astrocytoma. And so that's the classic example where patients have a overexpression of platelet-derived growth factor, which then in an autocrine loop results in the overgrowth of astrocytes producing an astrocytoma. So that's the first classic example of an oncogene mutation or a mutated gene, i.e. oncogene, resulting in formation or being associated with formation of a tumor. All right, so that's one example. Another example would be growth factor receptors. Another example, growth factor receptors. So again, we can go back to our diagram, and we can remind ourselves that if this is a cell, and the cell is expressing a growth factor receptor, right? Here it is expressing a growth factor receptor. When growth factor comes in, all right, if normally what would happen is growth factor would bind the receptor, signal would be sent, and the cell would then divide. However, if you've got way too many receptors, let's say there's a mutation and there's overexpression of this receptor, then a simple stimulus of growth factor is going to result in 1,000 times or 100 times signal to this cell to grow and divide, and so this cell will begin to grow and divide out of control. And that will eventually result in the formation of a tumor. Now, what are some important growth factor receptors that can become mutated, and what tumors are they associated with? Well, one of the very high yield growth factor receptors is HER2/neu. All right, HER2/neu is a growth factor receptor. It's an epidermal growth factor receptor that is amplified in a subset of breast cancers. And why is this important to be familiar with? Because it ties back into pharmacology. And you'll remember from pharmacology that there is a designer antibody called trastuzumab. The other name is Herceptin. But there is a designer antibody called trastuzumab which is designed against the HER2/neu receptor. And so patients who have overexpression of HER2/neu, i.e. amplification of HER2/neu, that results in breast cancer, in a subset of breast cancer, can be treated with trastuzumab. And there's a classic examination question in which you could be asked, a patient, I don't know, we can make up a question that sort of along the same principles. Patient comes to you having read, a patient with a history of breast cancer comes to you having read an article in the newspaper in which she sees that there is a new treatment for breast cancer that is designed against, that is a designer antibody against a growth factor receptor. So we could ask them, which of the following tests would be important to perform in order to determine whether or not she should be treated with the drug? A, estrogen receptor, B, progesterone receptor, C, HER2/neu, etc., etc. And obviously the answer would be HER2/neu. So it's important to be familiar with, and I would basically say that in a subset of breast cancer, there is amplification of the HER2/neu growth factor receptor. And so that's an important growth factor receptor to be familiar with. Another very important growth factor receptor to be familiar with is RET. And the key association with RET is medullary carcinoma of the thyroid, in particular medullary carcinoma of the thyroid associated with MEN2A and 2B. Now, this requires a little bit of discussion. So what's MEN? MEN is multiple endocrine neoplasia. It is a syndrome in which patients develop tumors in multiple endocrine organs, and it can be divided into MEN1, MEN2A, and MEN2B. And in MEN2A and 2B, one of the common tumors between each of those 2A and 2B is medullary carcinoma of the thyroid. Now, this is a very high yield association because if you suspect MEN2A or 2B in a patient, let's say a patient has some of the other features of MEN2A or 2B like pheochromocytoma and I don't know, mucosal neuromas, as would be seen in MEN2B, then those patients who in whom you suspect MEN, you can do RET studies. And if the patient has a RET mutation, then what you'll actually do is prophylactically remove the thyroid, i.e. the thyroid will be removed before tumor can actually form. And because medullary carcinoma of the thyroid could potentially kill the patient. And so testing for RET mutation is a very important feature of how we might deal with patients who have familial or MEN2A or 2B associated medullary carcinoma of the thyroid. So that actually is a very high yield association. And I'm going to give you much more detail when we do the endocrine lectures. I'm just again planting some seeds. Another important growth factor receptor is the KIT growth factor receptor, and a mutation in the KIT growth factor receptor is associated with something called gastrointestinal stromal tumor, and that's an important association to be familiar with. Okay, the next group of oncogenes are signal transducers, and the idea here is that after the growth factor receptor is bound, it will send a signal using a transducer to the nucleus. Now imagine for a moment, let's go back to our whiteboard, let's say that this is the cell, and we've got this receptor here. If growth factor binds the growth factor receptor, then a message will be transduced, and that message will go to the nucleus. Now normally, for one growth factor receptor molecule, you only want one growth factor receptor signal sent via the messenger. Let's pretend that the messenger gets mutated so that the messenger keeps sending signal to the nucleus to grow. If there's massive signal heading to the nucleus to grow, the nucleus will obviously cause the cell to divide, and multiple divisions will eventually result in the formation of a cancer. And so that's the idea in this particular subtype of oncogenes, and that is that if there are mutations in signal transducers, that'll result in the formation of a cancer. And so what are some classic ones to be familiar with? Well, the first and probably the most important is the RAS signal transducing protein. Why is this important? Because it is commonly mutated in a large number of cancers. In fact, I think up to 70 to 80% of human tumors actually have a RAS mutation associated with that tumor. So it's seen in multiple carcinomas, it's seen in melanoma and in lymphoma. So a broad range of human cancers actually have a mutation within the RAS signal transducer. This then requires us to go into a little discussion of biochemistry. So I'm going to take a step back here. This discussion is written in your book, but I'm going to go ahead and present it here because it's the perfect place in which to have this discussion. And so I'm going to simply go back and produce my whiteboard and just let you know a little bit about RAS. So what's the idea with RAS? Let's pretend that this is a cell, okay, and the cell clearly has a receptor, right, and growth factor would come in, and growth factor would bind that receptor. Now, when growth factor binds the receptor, one of the molecules that can be associated with the receptor is actually called RAS, R-A-S, okay? So that's RAS. Now, when RAS is associated with a receptor, it usually sits in its off state, which is basically RAS associated with something called GDP, all right? Now, the moment that growth factor receptor binds the receptor, the GDP gets kicked off, and instead, RAS gets associated with GTP. And when RAS gets associated with GTP, the RAS-GTP complex then can send message down to the nucleus. And that's the key